Okay, so welcome back to this next video in which we are discussing gastric acid secretion. Okay, so we're currently looking at how postganglionic parasympathetic cholinergic neurons from the vagus nerve can stimulate parietal cells to secrete more hydrochloric acid. Okay, so we've discussed that they will release acetylcholine onto the parietal cells, and that this acetylcholine will act on M3 muscarinic acetylcholine receptors. The G protein coupled receptor will then be activated, and it will interact with the GQ heterotrimeric G protein to activate the alpha subunit to alpha Q GTP. The alpha Q GTP subunit will then activate this enzyme here, phospholipase C beta, and I'll highlight this in pink here. And basically this is going to act on a target within the phospholipid bilayer called PIP2, which is short for phosphatidylinositol 4,5-bisphosphate. Okay, so we are discussing the structure of a normal phospholipid within the phospholipid bilayer so that we can then understand how PIP2 is basically just a modified phospholipid. Okay, so we've got glycerol being represented by this horizontal green line here, and this is a 3-carbon molecule, propane 1,2,3 triol, where you have alcohol groups coming off the first, the second, and the third carbons of that molecule. And basically you have esterified to the first and the second alcohol groups of the glycerol molecule long-chain carboxylic acids, and to the third alcohol group of the glycerol molecule you have esterified via a phosphoester link a phosphate group, basically. So this is a phosphate group. Okay, now basically the proper name, or the old name, for a molecule like this, a normal phospholipid, is to call it a phosphatidate molecule. And although people rarely do use phosphatidate to mean this, when we're talking about modified phospholipids such as PIP2, we use it all the time. This is what this phosphatidyl here means. It means one of these phosphatidate molecules attached onto something else, attached onto inositol. Okay, so I think we can probably squeeze in here the now structure of phosphatidylinositol 4,5-bisphosphate. So basically, to turn phosphatidate into phosphatidylinositol, all we need to do is bind the phosphatidate molecule to an inositol ring. Okay, so remember that the phosphate group, when you attach a phosphate group to an alcohol group via a phosphoester link, the phosphate group still has another alcohol group free. So if I get back this picture of serine being phosphorylated by a phosphate group here, we saw that the alcohol group of the phosphate group combines with the hydrogen of the alcohol group to produce water, and then the oxygen of the alcohol group binds to the phosphorus atom, okay, and that creates a phosphoester link. And basically the exact same has happened here, except that the alcohol group is now on the glycerol molecule rather than on a serine residue. Now, however, the phosphate group here has another alcohol group, and basically it still has one of these groups where we have the phosphorus atom doubly bound to an oxygen with an alcohol group coming off. It still has one of those intact, and this can basically link on to another alcohol group by another phosphoester link, and that's what's going to happen here. This other alcohol group of the phosphate group here is going to link to an alcohol group of the inositol ring here. Okay, so this 6-membered ring that I'm drawing here in blue, this represents inositol. Okay, and the proper name for inositol is cyclohexane 1,2,3,4,5,6-hexol. Okay, so cyclohexane, so that means it's a 6-membered carbon ring where all of the bonds between the carbons in the ring are single bonds. Okay, so it's cyclohexane 1, 2, 3, 4, 5, 6 hexol. Okay, so it's this 6-membered carbon ring where all of the bonds between the carbons are single bonds, and then off all of the carbons, you then have a single alcohol group. Okay, 1, 2, 3, 4, 5, 6 hexol means that every single one of these 6 carbons has an alcohol group coming off it, which overall then means that the whole ring has 6 alcohol groups coming off it. Okay, so that's the structure of inositol. So you have this 6-membered carbon ring where each of them has an alcohol group coming off. Now that still leaves each carbon with a free bond basically, and that will be to a hydrogen atom. Now it's a little bit more complicated than that, because if you actually think about this structure in your head, there are a huge number of different optical isomers. Okay, so if you imagine the 6-membered carbon ring as sitting within the plane, then each of the carbon atoms in this 6-membered ring has 2 more bonds coming off it. Now one bond will come out of the page towards us, and one will go into the page away from us. Okay, so either the hydrogen atom that comes off the carbon can come out of the page towards us, and the alcohol group can therefore go into the page away from us, or it can be the other way around. The alcohol group can come out of the page towards us, and the hydrogen can go into the page away from us. Now this leads to many different optical isomers of this molecule. Because, for instance, if you want to see this, an extreme example is that you could have all the alcohol groups on the same side, i.e. let's say they all go into the page away from us and all the hydrogens come out of the page towards us. Alternatively, you could have 3 on one side and then 3 on the other. So let's say we could put this alcohol group, and this alcohol group, or rather the alcohol groups coming off the carbons, these 3 carbons coming out of the page towards us, and then the alcohol groups coming off these 3 carbons going into the page away from us. That would be a distinct optical isomer of this inositol molecule. Okay, so there are many different optical isomers of inositol. There are actually 9 different optical isomers of inositol in total. Okay, and the form of inositol that's most common within cells is a form of inositol called myo-inositol. Okay, and the carbons in myo-inositol have a set naming system. Okay, and it turns out that the carbon that this phosphate group is attached to via a phosphoester link is actually carbon 1 of myo-inositol. Then this carbon here will say that's carbon 2, then carbon 3, then carbon 4, then carbon 5, then carbon 6. Okay, so we've now created phosphatidyl inositol. We've got phosphatidate molecules with inositol molecules linked off them, but we want phosphatidyl inositol 4,5-bisphosphate. So we want to stick a phosphate group onto the 4th and also the 5th carbons of this inositol ring. And again, these will be bound there via phosphoester links. Okay, and the reason I went to the lengths of explaining this optical isomerism here, okay, is to explain to you why this molecule is not called phosphatidyl inositol 3,4-bisphosphate. Because if you were going to just look at my cartoon and name this, you would think that this carbon here should be called 3 and this 4, okay, to minimize the numbers as much as possible, and then it should be phosphatidyl inositol 3,4-bisphosphate. But the reality is that we are dealing with a specific optical isomer of inositol in biological systems, okay? And that optical isomer has pre-named carbon atoms, basically, and the carbon atoms that the phosphate groups are added onto are the ones that are the 4th and the 5th. So basically, it's because of this optical isomerism that we have to be careful with the naming here. Okay, so all of that discussion was just to discuss this substrate of the phospholipase C beta enzyme. Okay, so PIP2 is the substrate for this enzyme, and hopefully what you can now appreciate is that basically it's just a modified phospholipid. It's just a phospholipid with a bigger head, with something stuck off the phosphate group that's dangling into the cytoplasm, okay, and this is a normal component of the phospholipid bilayer, okay, so it's normally within cells, cell membranes rather. Okay, right, and basically what's going to happen is that phospholipase C beta is going to break this phosphatidyl inositol 4,5-bisphosphate down into 2 other molecules. Okay, and what it's going to do is it's going to cut this structure between the glycerol molecule and the phosphate group here. Okay, and it's therefore going to create two products, basically. One of the products is going to be this molecule where we've got glycerol with two long-chain carboxylic acids linked off it. Okay, like so. So here's the glycerol molecule in green, and here are two long-chain carboxylic acids which are esterified to the first and second alcohol groups of the glycerol molecule. Okay, now this molecule is called diacyl and then glycerol. So diacyl glycerol. And for short, diacylglycerol is abbreviated to DA for diacyl and then G for glycerol. Okay, and another name for diacylglycerol is just to call it diglyceride. Okay, so there are these two names, diacylglycerol or diglyceride. Now, I'd just like to explain to you why this is called diacylglycerol. So the glycerol should be pretty common sense. We've got a glycerol molecule here. But why diacyl? What does acyl mean? Well, basically, if we look back up at this diagram where I showed esterification, and we were here talking about the esterification of acetic acid to choline, but the principle is the same. Imagine that this is now the alcohol group of a glycerol molecule, and this is some long-chain carboxylic acid. Basically, what you can see is that you don't add the full carboxylic acid molecule on. Okay, you take this alcohol group off the carboxylic acid molecule and then add what's left on. And this portion that is left here, okay, and this is of a generic carboxylic acid rather than specifically acetic acid. Okay, for a generic carboxylic acid, this is called the acyl group. So imagine that instead of having a methyl group specifically here, you have just some arbitrary group. Okay, it's called the acyl group of a carboxylic acid. Okay, so that's what we've actually added onto the glycerol molecules. We've added two acyl groups on, and that's why it's called diacyl, because you've got two acyl groups added onto glycerol. So diacylglycerol. Okay, and the diacylglycerol molecules remain attached to the inner leaflet of the phospholipid bilayer. Now, the other product is what's left here, which is the inositol ring with free phosphate groups coming off it. Okay, and this will not remain bound to the phospholipid bilayer. So let me just draw it like this because I'm running out of space. So I'm just drawing it at a bit of an odd angle. Okay, like so. This will go off into the cytoplasm, okay? So remember, all that was attaching it to the phospholipid bilayer was the fact that it was previously attached to this diacylglycerol molecule, which was attached into the phospholipid bilayer. So once it's freed of that, it will float off into the cytoplasm. Okay, and the name for this molecule is inositol, because we've got an inositol ring. And then we've got phosphate groups coming off the first, the fourth, and the fifth phosphate group. So inositol 1, 4, 5, and then it's trisphosphate because we've got 3 phosphate groups. Okay, and again, the reason this isn't called inositol 1,3,4 1,3,4 trisphosphate is because there is a specific naming system for this optical isomer of inositol, and some people who are being really strict would call this myo-inositol 1,4,5 trisphosphate. So occasionally, if you read the research literature, you might hear it called myo-inositol 1,4,5 trisphosphate. Okay, but basically that's just referring to the specific optical isomer of inositol that is contained within it. And for short, inositol 1,4,5 trisphosphate is usually abbreviated to I for inositol, then P for phosphate, and because we've got 3 phosphate groups, you subscript it with 3 to create IP3. Okay, so the two products of phospholipase C beta activation are that you get diacylglycerol molecules and inositol 1,4,5 trisphosphate molecules. Okay, now, basically, these together are going to contribute to the activation of an enzyme called protein kinase C. Okay, so let's go over onto the other side of the page. Okay, so to activate protein kinase C, you need two signals. Okay, now diacylglycerol is one of these signals. So here comes the diacylglycerol molecule. Okay, so we'll have this as our long chain carboxylic acids again, and we'll have our glycerol molecule in green. So this is our diacylglycerol molecule, and basically it's going to stay in the inner leaflet of the phospholipid bilayer, and it's going to activate an enzyme that is close to the inner leaflet of the phospholipid bilayer here. Okay, so this enzyme is protein kinase C, or PKC for short. So this is protein kinase C. Okay, now to activate protein kinase C, one of the things you need is diacylglycerol, but you also need calcium. And calcium is going to be released from the ER by inositol 1,4,5-trisphosphate. So let's draw our other product here now. Okay, so here is the IP3. So we've got our inositol ring, this 6-membered carbon ring here. And then we've got phosphate groups coming off the first, the fourth, and the fifth carbons of the myoinositol ring. Okay, and basically this will diffuse into the cytoplasm, and it will find the intracellular organelle known as the endoplasmic reticulum. Okay, so the endoplasmic reticulum is a membrane-bound intracellular organelle that, amongst other things, serves as an intracellular store for calcium ions. Okay, so within the lumen of the endoplasmic reticulum, you have very high calcium. Okay, so this is the endoplasmic reticulum, and for short, the endoplasmic reticulum is often just abbreviated to the ER for endoplasmic and then reticulum. Okay, now in the ER membrane you have special receptors for IP3, which are called IP3 receptors. Okay, so I'll colour this in turquoise here. So this is an IP3 receptor sitting within the ER membrane, and these IP3 receptors are basically calcium channels which can allow calcium to move from the lumen of the ER into the cytoplasm. Okay, now usually they are shut. When IP3 levels go up in the cytoplasm, it leads to the opening of these IP3 receptors. It's not direct, okay? IP3 doesn't just directly bind here and they open. It does bind to them, but that doesn't directly cause their opening. But for a decent approximation, it leads to them opening. Okay, so we'll settle for that. IP3 leads to the opening of these IP3 receptors, but not directly. It does directly bind, but it gets them into a state where they can then be activated to open. Okay, and if you're more interested in that, do please watch my videos on calcium signaling. Okay, so what will then happen is calcium will be released from the ER, and the calcium will then go and do the second part of the activation of the protein kinase C. So calcium will bind to protein kinase C, and then the protein kinase C will become active. Okay, and now what does protein kinase C do once it's activated? Well, it does a very similar thing to protein kinase A. It's a serine-threonine kinase, and again, it will phosphorylate the same target as protein kinase A. It's going to go and phosphorylate the proton-potassium ATPase, which, remember, drives hydrochloric acid secretion. Okay, so it has different phosphorylation sites to protein kinase A, but the effect is the same, that you activate the proton-potassium ATPase, and then the proton-potassium ATPase will secrete a greater number of protons per second, let's say, and since that drives the secretion of the hydrochloric acid, you'll therefore get an increase in the secretion of hydrochloric acid. So overall, the postganglionic parasympathetic cholinergic neurons of the vagus nerve, these stimulate parietal cells to increase hydrochloric acid secretion by activating protein kinase C within the cytoplasm of these parietal cells. Okay, so that now concludes our discussion of gastric acid secretion.